Hello, my name is Isabelle Husted, and today I will be talking about characterizing the phenology and fecundity of Spartina alterniflora in Georgia coastal salt marshes. This research was completed with the help of Alexander Gregory and Hannah Riley under the guidance of Dr. Heather Joesting in the Department of Biology at Georgia Southern University. Salt marshes provide valuable ecosystem services such as erosion control, protection from floods and storm surges, maintenance of commercial fishery species, and carbon sequestration. However, as much as 50% of salt marshes have been lost or degraded globally. Fortunately, in recent years, efforts to restore these marshes and habitats has increased. That brings us to our species of interest, Spartina alterniflora. This is the dominant species of cordgrass in our salt marshes that plays a valuable role in maintenance and growth. For this reason, Spartina is the plant most often used in salt marsh restoration projects. Our research sought to characterize the phenology, the reproductive status, and fecundity, seed count, viability, germination, and survival rates of the smooth cordgrass, and to see if marine influence has an impact on those attributes. We visited 3 sites of varying marine influence and salinity. Lazaretto Creek Boat Ramp had the highest, Bradley Hall Boat Ramp was next, and Halcyon Bluff Clubhouse had the lowest. For fecundity, we collected mature spikelets from all 3 marsh sites, counted the seeds, and placed them in cold, wet storage for approximately 3 months to break dormancy. After stratification, we tested 25% of the seeds from each spikelet for viability using a tetrazolium chloride test. Any spikelets with 5% viability or greater were used for germination trials. After successful germination, plants were moved to the greenhouse and monitored for survival. However, I should note that germination rates are unclear due to the COVID-19 shutdown in 2020. This disrupted the trials for the 2019 seed collection. We found that the site had significant impact on seed count and viability, with Lazaretto Creek and Halcyon Bluff having the most. There was no significant difference in fecundity for sites between the 2 years, except for the Halcyon Bluff, which had significantly more seeds viable from the 2020 collection compared to the 2019 question. For the phenology, methods included laying a transect in the upper and lower marsh and measuring all individuals of quadra every 2 meters. The result of this was at the height of reproductive status, 25% of plants sampled were reproductive. The best time for flowering was early October, the best time for fruiting was early to mid-October, and the best time for seeding was mid to late October. The conclusion from this is that the best time for seed collection would be mid to late October. More data is needed to determine if there is a marine influence on the phenology, and there was no significant difference among sites in germination success and survival. Thank you, and I hope I taught you something today.